The immune response in inflamed tissue, such as in autoimmune target organs, is in an unbalanced situation between the tolerogenic and the immunogenic cells. In this condition, immunogenic dendritic cells are able to secrete pro-inflammatory cytokines such as IL-12 and TNF-alpha, and are able to stimulate CD8 T cells and Th1, Th17 T cells to produce IL-17 interferon gamma and to have a damage of the tissue. It is known that to restore the tolerance condition, we have to induce regulatory T cells, dampen the presence of effector T cell, or inducing a specific condition in this effector T cell named as exhaustion. So one of the major players in these games are the tolerogenic dendritic cells that are able, through the secretion of anti-inflammatory cytokine IL-10 and TGF-beta, to induce and expand tier 1 regulatory T cells and control the effector T cell response and the balance between the tolerogenic and immunogenic cells. During years, different strategies to generate tolerogenic dendritic cells in vitro have been exploited, both from the bone marrow precursor and from the CD14 monocytes, through the use of the differentiation of these cells in the presence of anti-inflammatory cytokines such as the IL-10 TGF-beta, immunomodulatory molecules such as vitamin D3 and dexamethasone, and immunomodulatory drugs such as rapamycin. Moreover, it has been exploited the possibility to prevent the maturation of these cells by the use of short RNA against the co-stimulatory molecules of the dendritic cells. All these strategies arrived to clinics and have been applied in different autoimmune diseases such as the type 1 diabetes, the rheumatoid arthritis, and the multiple sclerosis. Even our lab focused our attention on a different strategy to generate tolerogenic dendritic cells in vitro, and we focus our attention on by the genetic modification using lentiviral vectors. So the aim of my project was to define the mode of action of genetically modified tolerogenic dendritic cells, and then we want to assess the efficacy of these genetically modified tolerogenic dendritic cells to restore or promote the tolerance in preclinical model of type 1 diabetes. So we generate antigen-specific control dendritic cells by transducing dendritic cells with a vector encoding for a specific antigen, and with this strategy we want to elicit an effector T cell response. And then we want to generate two different tolerogenic dendritic cells. The first one, it is obtained by the transduction of dendritic cells with a vector encoding for the antigen coupled with multiple target sequence of miRNA 155-146A. This strategy will allow the presentation of the antigen in an immature state, and this will promote T cell energy and tier 1 T cell differentiation. And the second strategy that we want to use, we want to couple the expression of the antigen of interest with the anti-inflammatory cytokine IL-10, and this will promote again T cell energy and tier 1 T cell differentiation. So, starting with the first aim, we want to be sure to present the antigen both in the context of class 1 and class 2. So to do that, we couple the expression of the antigen with the invariant chain. With this strategy, the antigen will be presented in the context of class 1 as an endogenous antigen to elicit CD8 T cells. At the same time, the presence of the interaction of the invariant chain will allow the presentation and the process and the presentation of the antigen in the context of class 2 as an endogenous antigen to elicit the CD4 T cell response. So we verified that our strategy is feasible, and we generated two lentiviral vectors. The first one encodes for a short sequence of the OVA peptide, of the OVA protein that is specifically recognized by OT2 CD4 T cell, and the second vector that encodes for the longer sequence of the OVA that is specifically recognized to both CD4 and OT1 CD8 T cells. So we generated our dendritic cells and we stimulated both OT1 and OT2 cells. As you can see from this graph, we confirmed that our strategy is feasible. So the cells that are transduced with a short sequence of the OVA are able to elicit only a CD4 T cell response of the OT2 OT2 here in red, while the cells that are transduced with the longer sequence of the OVA are able to stimulate both OT1 and and OT2 T cells. So then this is the experimental protocol that we used to generate our engineered dendritic cells. We started from total bone marrow at day 0 in presence of GM-CSF. At day 2, we performed the transduction with control lentiviral vector, Mirna-regulated lentiviral vector, or the bidirectional lentiviral vector. And at day 8, we have our cells, and we have immature cells, or we can activate it, then obtaining a mature-like phenotype by the adding LPS in the culture. So then we analyze our cells from the phenotypic point of view, from the cytokine production profile, and we analyze the modulation of the cells to both CD4 and CD8 T cells. So first of all, when we characterize our cells from the phenotype point of view, what we notice is that the dendritic cells transduced with the IL-10 vector show downregulation of the expression of the CD11C and the class 2 compared to the other cells. But more importantly, our cells show cytokine profile secretion that is similar to the one of the tolerogenic cells. So the IL-10 transduced cells have a secrete high amount of human IL-10, which is encoded in the vector, and low levels of IL-12, of the pro-inflammatory cytokine IL-12. Moreover, we notice a reduction in the level of IL-1 alpha, IL-6, and TNF-alpha, which was not statistically significant compared to the groups. Then we move to the first strategy of our tolerogenic dendritic cells. So the approach is always the same. We differentiated our cells transduced with the vector encoding for the OVA and multiple target sequence of Mirna 155, 146A, and at day 9 we stimulate our target cells. I just want to focus your attention on this strategy. So these two microRNA are known to be upregulated only when the cells are in a mature-like phenotype. So when we transduce the immature cells with the vector encoding for the Mirna target sequence, we obtain a similar level of proliferation of both CD4 and CD8 T cells compared to control. When we stimulate our target cells with mature-like cells transduced with the Mirna, the post-transcriptional regulation of the Mirna target sequence is activated, and we downregulate the proliferation of both CD4 and CD8 T cells. Then moving to the second strategy of that we want to use for the tolerogenic dendritic cells, we transduce our cell this time with the bidirectional vector encoding for the OVA and the IL-10, and we stimulated both CD4 and CD8 T cells. So what we notice is that for the CD4, when we stimulate the cells with the IL-10 vector, both at immature or LPS activity status, we notice a reduction in the proliferation of the CD4 T cells. While when we stimulated the CD8 T cells, we have a reduction of the proliferation only when we stimulate the CD8 T cells with the dendritic cells at mature-like state. Then move to an in vivo experiment to put us in a more physiological condition, and we generated chimeric mice with only 5% of OT2 cells. At full reconstitution, we performed 5-week injection of our dendritic cells, transduced with the OVA, with the OVA-10 and the PS control. After 3 weeks, we sacrifice the mice. And what we notice is that we have an expansion of antigen-specific T cells in the mice treated with the antigen. And if we look in the T-Rex compartment, we do not have any difference in the FOXP T-Rex compartment, while we have an expansion of tier 1 T cells in the mice receiving the DCIL-10 transduced cells. And moreover, when we re-challenge the splenocyte of those mice with, again, the antigen, the mice that receive the tolerogenic cells are hyper-responsiveness compared to controls. In a similar way, we generated chimeric mice with only 5% of OT1 T cells. At full reconstitution, we performed 5-weekly injection of our dendritic cells transduced with the OVA, IL-10, OVA-MIRNA, and DCGFP. And according to the in vitro data, we injected cells both at immature or LPS activity. At week 17, we sacrificed the mice. And what we notice is again an expansion of the CD40 of the antigen-specific T cells that in mice that receive in the ova, and more importantly, we notice an expansion of the CD8 T cells that express the PD-1 lactate-3 marker. Marker indicating that the mice that receiving the IL-10 treatment show an exhausted phenotype in the CD8 of those cells. So to conclude this first part, I showed you that the dendritic cells transduced with the IL-10 vector are phenotypically similar to control disease, but express low level of CD11C and class II molecules, and show a cytokine profile that is similar to the one of the tolerogenic dendritic cells with IL-10 and LOAL12. That upon activation, that MIRNA 155-146A is able to post-transcriptionally regulate the presentation of the antigen. And moreover, that the IL-10 both steady state upon activation inhibit over-specific CD4 T cell proliferation, and only activated dendritic cells transduced with the IL-10 are able to modulate the CD8 T cell proliferation in vitro. In vivo, what we observed is that the dendritic cells transduced with the IL-10 are able to promote over-specific tier 1 T cells and CD8 T cells that show an exhausted phenotype. So moving to the second aim of the project, in which we want to assess the efficacy of these genetically modified tolerogenic dendritic cells, we generated antigen-specific control disease and only tolerogenic dendritic cells transduced with the IL-10 encoded vector. In the type 1 diabetes, it is known that the insulin B is the main autoantigen involved. It is not the only one, but it's one of the most important. And recently, it was discovered that the immunogenic peptide of the insulin with a single substitution with the R32E increase a lot the affinity of the NOD class II molecules, generating a packet that is called insulin epitope. And more recently, it is also discovered that in the pancreatic beta cells, both in the human and in murine context, modification of autoantigens have been generated, generating hybrid insulin peptides that are formed by a portion of the insulin and a portion of other related peptides, such as the chromogranin A. These peptides are specifically recognized by specific T cell clone, and one of them is the BDC 2.5. So we move in vivo and we set up two different induced models of type 1 diabetes. In the first one, we transfer the transgenic CD4 T cell from the BDC 2.5 mice in NSG mice, and we monitor the blood glucose level, and we have the onset of type 1 diabetes in 10 days. And in the second model, we transfer a total splenocyte from diabetic NOD mice in NSG mice, and in this case, we have the onset of the type 1 diabetes in 25 days. So first of all, we verified in vitro with the same approach that our cells are able to, that our transduced cells with the IL-10 able to control the antigen-specific T cells, and this was the case for both of the models. So reflecting the results we obtained for the OVA, the CD4 T cells show an hyperproliferation compared to control, both in the BDC 2.5 and in NOD context. So moving in vivo, in the first model, we injected our effector cells, our diabetogenic cells, and then we perform multiple injection of 3 million of dendritic cells, and we injected dendritic cells transduced with the antigen, in this case is the HIP, or the antigen with the IL-10. And results show that the mice treated with the dendritic cells with IP have an acceleration of the disease compared to controls, while in the mice injected with the dendritic cells with the IL-10, we have 50% of the mice that are protected from the disease. In the second model, the strategy was very similar. We injected our diabetogenic cells, and we performed a weekly injection of 3 million of dendritic cells transduced with the insulin IL-10, or as a control, we used dendritic cells transduced with the IL-10 and the OVA as unrelated antigen. And results show that all of the mice treated with the non-tolerogenic cells show a similar behavior with the control dendritic cells, while we have 40% protected mice in the group treated with the IL-10 and the insulin. So overall, we can conclude that the lentiviral IL-10 antigen platform allows the generation of tolerogenic dendritic cells that show a cytokine profile of the tolerogenic dendritic cells that is able to inhibit antigen-specific T cell activation and proliferation in vitro. And in vivo, I've showed you that the repetitive administration of engineered dendritic cells are able to promote antigen-specific TR1 and also to lead antigen-specific CD8 T cells with an exhausted phenotype. Moreover, it are able to prevent type 1 diabetes onset in approximately 50% of the treated mice. So we strongly believe that this platform can represent a promising tool to engineer dendritic cells for a cell-based approach to restore tolerance in T-cell mediated diseases. So finally, I want to thank my DOS, Silvia Gregori, all the lab members, my second supervisor, Kari Ilkins, the tutor of the UNISR, Marta Muzio, and all of you for your attention.
Thank you very much, Laura. Can you remove your presentation? And the thesis is open for discussion. Ruggero, Filippo, do you have any question?
I would give Filippo the right of way. He's been waiting for. Filippo, do you have?
You're muted,
Okay.
Filippo.
Sorry. No, actually, I have no question at this time. So for me, sorry, but.
So you are satisfied with the thesis?
Yeah.
Ruggero?
I'm also very happy, and I have no questions or comments.
Same for me, Laura. It was really well presented, clear, and the topic was a great topic. So thank you very much for your presentation. Thank you. We can switch to Italian now because the presentation part is ended. Adesso la procedura è che io e il professor Pardi e il professor Casoni ci riuniamo in un'altra aula